In a previous video, I showed you a tool we built to study criticality across frequency bands in the human brain. It revealed striking differences between the brain's harmonies in healthy participants and those with disease. Today, we are adding a second lens from physics, the renormalization group, the math of zooming to see what stays the same across scales. Curious? Then stick around. So, what are we working on right now? In short, how the dynamical behavior of the brain changes when you look at it with a different time resolution. We first scanned brain activity across frequencies and saw patterns that looked like a system on the verge of a phase transition. Think water just about to boil. Now we're asking, do the signatures of criticality hold up when we zoom in and out in time? That matters because brain data comes in very different time scales, from millisecond spikes in single neurons to local field potentials unfolding over tens to hundreds of milliseconds, all the way to slow oxygen level signals that take seconds. We are testing whether the same near-critical behavior shows up across all these time resolutions. To test this, we use a classic physics tool called the renormalization group, basically the math of zooming. It asks, how do the rules of our universe change when you change the resolution? That's why everyday motion looks Newtonian even though deep down nature follows quantum physics. When you zoom out, messy microscopic details get averaged away and only the important stuff survives. You don't need the speed of every molecule to know a room's temperature, you just need the collective behavior. It's a bit like art. If you press your nose against a Van Gogh, you see brushstrokes. Step back and the picture will emerge. In statistical physics, this tool also tells you how close a system is to a phase transition. At a critical point, something special happens. The system becomes scale invariant. Zoom in or out and the pattern looks the same. That's exactly the signature we're hunting for in brain activity at different time resolutions. The simplest way that we zoom out with the renormalization group is by averaging neighbors, what physicists call coarse graining. Think back to the Van Gogh example. If you squint, the brushstrokes blur and the composition appears. We do the same with data. We take a small region, combine the values into one and then repeat. The fine details fade and the large scale pattern remains. Well, what's the catch? In the brain, distance is not equal to similarity. Two far apart neurons can fire in synchrony, while neighbors may do totally different things. So, instead of averaging by where cells are, we average by how they behave. We group together signals that are highly correlated, they rise and fall together, and coarse-grain those. This behavior-based zooming is the essence of the phenomenological renormalization group. We applied this behavior-based zooming to neural activity right on the edge of a phase transition. Inside each group of neurons, we tracked three things. First, how the main activity patterns stack up, the principal components. Second, the overall size of the fluctuations, the total variance. And third, how long the activity stays similar before changing, the correlation time. All three followed clean scaling laws with the size of the coarse graining. The principal components fell off as a power law with exponent mu, the variance scaled with alpha, and the correlation time with zeta. In physics, those exponents act like a system's fingerprint. They label its universality class. Now here's the twist. We showed both on paper and in simulations that these exponents aren't fixed. They shift with the time resolution you use. Change how much you coarse grain in time and you get different critical exponents. In other words, spike trains and oxygen level dependent signals don't exactly follow the same scaling laws. What surprises most is the tight mathematical link between time resolution and the inputs a neural network receives. Neural circuits turn stimuli into information, but we found that how finely you look in time is equivalent to changing the temporal structure of those inputs. Using a finer time resolution acts like driving the network with inputs that change rapidly with a short correlation time. Coarse graining in time is equivalent to inputs that vary slowly, with a longer correlation time. Think back to the painting. That's our input. Now imagine two cameras. One is perfectly sharp, the other blurs. That's our coarse graining. Here's the point. A blurry painting photograph with a sharp camera is equivalent to a sharp painting photograph with a blurring camera. Same image, different sources of the blur. Since input statistics can strongly shape the critical exponents that we measure, we had to ask, when experiments report near critical behavior in human brain data, how much is truly intrinsic to the brain, and how much is inherited from the temporal structure of the inputs? So, we stress-tested the idea on the simplest network possible, neurons with no connections, each one independent. We drove this network with inputs whose correlation time we could dial from fast to slow, and we mimicked real experiments by analyzing short recordings on the order of minutes. To our surprise, this uncoupled system produced the same scaling exponents mu, alpha and zeta as a network genuinely near a critical point. Translation: input statistics and finite recording windows can mimic near critical fingerprints. I'll be honest, this kind of freaked me out. I'm doing my thesis on the critical brain hypothesis, and for a minute I thought, great, so everything I did was wrong. Well, yes and no. First, what was off? I've been working with fMRI datasets, which are about 15-minute recordings with time series showing around 10 seconds correlation time. With that combo, theory says that you're in the danger zone. Finite windows plus long correlation time can fake criticality. So I ran a sanity check. I randomized the data in time, basically shuffling the time series. That keeps the temporal structure of the data but kills all possible correlation among pairs. If the critical signatures survive that shuffle, they're likely spurious. I did this and drumroll please. Yep, I get the same results after reshuffling. Luckily for me and my thesis, that wasn't the end. By treating each participant as an independent realization of the same dynamics, I could pool samples. 50 minutes per person across 136 participants took me about 34 hours of data. That pushed us well outside the danger zone. When I randomized this pooled dataset, the critical signatures vanished, which means the original weren't the spurious. Well, what's the takeaway? With fMRI, you usually can't make strong claims about criticality at the single participant level. The time series just isn't long enough. So when you see a paper doing that, be skeptical. Remember, even independent processes with long correlation times can mimic critical behavior.